also the microfluidic platforms for cancers and other diseases. And his innovations have been licensed to a lot of companies, and several of techniques have been approved by the FDA. So let's welcome Professor Utkan.
Thank you very much. Thank you very much. Yes. Okay, does it work? All right. Which one is which though? All right, great. I would like to start by thanking the Teresek Institute, Stewart and Ali for giving us the opportunity to, again, come out of the corners of our offices and meet some of our old friends, colleagues, and get an opportunity to make new friends. And I would like to start my talk acknowledging to hundreds of thousands of people that have lost their lives in 2 unfortunate earthquakes in Turkey in the past month, which are one of the deadliest events that has happened, I guess, over the past century within Turkey. Not the deadliest for sure if you think about the events in the Second World War, but I just want to start by acknowledging that and giving my condolences. Now, I have a talk that covers quite a few areas, and I just want to, at the beginning, disclose that some of the work that we have done and I'll show has led to different startup companies that I have co-founded or licensed out from my lab, so keep that in mind. And these are the areas that I'll try to cover, starting with telling you about what we are doing at the Canary Center for Early Cancer Detection and how the technologies that we are trying to develop as a group, and then more focusing, of course, the work that I have done in my lab, impact medicine in general, going through different areas and tying it back to the cancer at the end. So Canary Center, just to give you a background, was founded by late Dr. Sam Gambier and Don Listman more than a decade ago as a vision to bring cancer early detection into reality. And the idea was a very simple mission statement to focus on localizing and detecting cancers early. Today we imagine with everything that's happening with GRAIL and others, this has become really a high impact area. But it's going to continue to grow, and even some of the vision in 10 years from now, could we even predict cancers and curb them? It's some of the work that that's going on also at Stanford at the PHIND Center, the FIND Center, and thoughts around monitoring disease before that happens. Just to kind of walk you through this quick table, and I have another slide on this, especially in the aggressive cancers. If you look at pancreas and liver cancers, if we can actually detect cancer early, we can really increase the chances by many-fold of 5-year survival. So that's a very clear statistic from the Sears network. And just to kind of put it on the perspective, pancreatic cancer being one of the deadliest cancers, which is really important to detect and hard to image or reach. I think the importance of what we can do and how we can change medicine by bringing these new tools, technologies, instruments, ideas forward is a great mission. And by no means we don't underestimate the challenge. This is really a big problem, not only to detect, but to detect specifically, and then to bring in imaging and other modalities to localize. I think all those combined, we'll see a really interesting future for this. And we try to not only do it today with people that we have, and we try to kind of prepare the future generations of cancer research through NIH-funded programs. This is a program that has been going on more than a decade. We are just getting renewed for an NIH R25, as I saw the JIT this morning. It's a pleasure to be able to train undergrads and then prepare them for, in those young brains, for working in this field in the future. That is a huge impact. My lab, did I skip a slide? My lab mainly focuses on finding a real need in medicine and then trying to innovate around it, always keeping in mind what Da Vinci said. We are trying to look for simplicity, because what I've learned over the years, it is harder to make simpler devices that work, but it's much easier to translate those devices when they come into reality. So I'll talk about, I guess there's a delay, yeah. I'll talk about some of the work that we have been doing, looking at devices and scale. And when I say scale, you know, nanoscale, microscale, microfluidics, and how they actually have a different approach, and how realizing that has led to different applications that came over the past decade out of my lab. And this is something that I kind of present because it so nicely depicts an imaginary future, something Feynman stated so nicely. There is so much more at the bottom in the nanoscale. But I really like this visualization that we could have these small nano and micro robots that could go and fix things for us. And some of that came across in longevity and other discussions in the earlier days of this meeting. But when one looks at scale, it becomes important to understand that the physics, dynamics, and hydrodynamics, especially in the microfluidic space that I am in, changes quite a bit. And this is how far a frog can jump versus an elephant can, thinking about the scale. And when you kind of transcend this from meters all the way to micro and nanoscale, all the rules are different. And that realization led to a very simple but interesting tool that has led, I would say today, more than 100,000 babies born with IVF in sperm selection. And the question was, how do we select and how do we understand sperm as a cell? It's the fastest cell, and it's got a lot of mitochondria and nano engines where the tail goes into the cell body, and it makes these sinusoidal and kind of circular corkscrew movement, and understanding that movement not in a large drop but in a small scale, like it kind of follows these channels on the vaginal wall over a 5 to 7 day progress can lead to new devices for us to pick the right sperm in IVF. So I'll try to go through because I have a lot of slides, but one thing to realize is sperm has a lot of morphology, and we can definitely put it under a microscope and select for the best looking, best morphology sperm. But we can't predict if there is DNA fragmentation or if that sperm is going to lead to an embryo all the way to the blastocyst stage that can be actually, transferred with embryo transfer and leading all the way to a baby, a healthy baby born. When we look at different factors that affect us, these environmental effects, including air pollution, which is a big problem in the world, they all bring these reactive oxygen species, and they lead to, oops, there's a delay, it's amazing. Oh, sorry about that, guys. Going back. It leads to DNA fragmentation. And DNA fragmentation, as I said, is something that you would have to stain the sperm cell for, but if you stain it, then you can't use it. So is there any indirect way to kind of predict these damages? And just ibuprofen can lead to damage on the cells. There are things that we cannot predict today that we can see damaging the cell environment, cell DNA fragmentation. And when you look at nature, and Jeff, I think, spoke to this a little bit, when you look at nature, you get about 10 million sperm, and they, over a 5 to 7 day, only 1,000 to maybe 100 to 1,000 of them make it all the way to the egg, and 1 or 2 of them would actually create the embryo. So that's a big, long journey. And the journey takes place, in the reproductive system. If you look at millions of years of evolution of the reproductive tract, it's been preserved. So there must be a logical reason for nature to keep that process. And when you look deeper, for instance, in bovine, you see that these channels are on the walls that the sperm actually can go and guide itself through. Excuse me. If you look at what's happening today in the clinic for couples, there are about 75 million infertile couples in the world. We have IUI systems where you kind of wash the sperm and put it all the way into uterus, or something that Dr. Jean-Pierre Palermo came up with, which is called ICSI, intracellular cytoplasmic sperm injection, and you can take an egg and the sperm and inject it with a needle. And ICSI has really changed the game for IVF. However, from a surgical procedure to collect the eggs, you get about 10 eggs on average, and you have thousands and thousands, maybe millions of sperm to select from. And you have 1 egg, 1 sperm. Which one do we select? The standard approach about 10 years ago when we started this research was centrifuge it, smash all the sperm down, which created additional reactive oxygen species, additional DNA fragmentation that was known in the literature, and then see which ones swim up, and the embryologist would go and just pick one of the sperm. They would say, this is it, because of my experience with years, I can pick the right one. But when you go and ask actually what they are looking for, they were looking for morphology, they were looking for sperm that can swim longer straight. So we kind of took those and started studying sperm with Dr. Erkan Tüzel, who was at WPI at the time. He's a physicist. And we realized that when we put sperm in a droplet, they go all sorts of directions. There isn't really some hyperactivated ones on the right top. But when you start actually putting them into microchannels, and especially in microchannels where you can control the periodicity and hydrodynamic microenvironment, you started seeing some of the sperm directly going to the end, and some of them getting lost. And when you dig deeper to understand how this might be working, we realize that by simulating sperm and all the black dots around it is like water molecules, you can imagine, putting it into a very closed channel. Sperm's about 50 microns, including the tail length. If you put it somewhere below 200 microns, where the hydrodynamics are significantly changing, then we notice, and this is simulations that run a couple days, that start showing you that sperm is not just swimming, but the waves that it's creating in that microscale and the waves it's receiving that's bouncing back from surfaces is enabling a synchronization. So there is a certain geometry to how sperm swims with it's interacting with its microenvironment, and it's a hydrodynamic phenomenon. By changing or picking sperm that is a pinhead, meaning very small head, or a globospermic sample, which is common with a large head, you can see that these cells can get lost into this passive trap, but the ones that are really good morphology and able to swim make it to the end of the channel. And this is almost, I give the analogy of light hitting the surface of a lake, either bouncing back, depending on its frequency, or kind of going inside the lake or traveling parallel. So you can almost create a bandpass filter for electrical engineers in the crowd to control what passes. And this became an FDA-approved product, and we have been actually since 2018, it's been in use in about 45 states now, and more than 400 clinics last time I checked. So the idea is among thousands of sperm to pick the ones that are best swimmers and that fit the morphology of the best morphology to be able to go through this terrain. And by the time the two sperm that we picked and particle tracked on the left, by the time they reach the end, within 10 minutes, the one that's swimming very fast on the front will get there, and many others that are good will get there. And the advantage is these sperm that make it to the end, that has the right mitochondrial micromotors near the tail, they don't produce as much ROS when they are doing this process. So it's a marathon, as I explained in the nature. So that means the sperm that we select at the end are low DNA fragmented sperm. And that's the interesting part. I'll skip through some of the clinical studies. Now there are, I think, thousands of studies on this with very interesting papers. Just to highlight some of the early ones was done by Dr. Mitch Rosen of UCSF, who looked at the DNA fragmentation before and after sorting and compared it to a DFI, density gradient centrifuge that they had, which is the best method of the state of the art, so to speak, at the time. And they showed that the DNA fragmentation reduces, oops, pressing the wrong button all the time, reduces below a couple percent, even if you start with a sample that's 30% DNA fragmentation. And this becomes more pronounced for really high DNA fragmentation male, especially 50% of the population is male infertility. And if you have DNA fragmentation above 30%, that's considered to be a reason for severe male infertility. And by just running this through, and this was published in Human Reproduction, and it kind of made a difference. So they started, since they cannot predict who has, especially about 10% of the cases are fertility that is unexplainable. They call it idiopathic cases. So they kind of were suggesting that all the cases should use this system, because you're not doing an hour centrifuge, you're just going ahead and loading the chip with a single raw semen without any processing, coming back after a half an hour of incubation and using it. So that efficiency is great, which made it exciting in COVID, where most of the pickup of the technology by the market happened, and I can come back to it at the end. But there were other studies that looked at the embryo quality. I mean, we don't expect to change all the way to the end. What's going to happen when we reach the live birth rates, but we can change out of those 10 eggs. If we can have extra few embryos or embryos with better quality, that is then directly relating to the success of the implementation and so forth. So this is one study that was showing that they had better grade A or grade 1 quality embryos using the system on a randomized study. What I'm kind of seeing most recently, and I didn't include it here, it was showing that it was impacting the euploidy rates of the embryos, meaning you were getting genetically better and usable embryos, which is, I think, would be a showstopper, a great result for this to even push it forward. But you know, once you pass it to clinicians, there will be thousands of more studies, larger studies that come out. One thing that we have seen is in all the studies that it's not doing damage, but it's bringing advantages, especially for cases where there's male infertility or male factor infertility. Another area that I saw impact, and this is more going into socioeconomic impact, was you can, when a couple in a small village, let's assume a poor farmer's village, somewhere in a developing area, or even in the US, right? We have areas like that. They have two options: to go to the doctor to do IUI, meaning sperm is washed and injected into the uterus, or they go and do this $50,000 ICSI process. And they always try twice the IUI just because of the, the rule book, but it's not considered to be successful. We showed that by standardizing the IUI process with sperm washes, we are enabling little clinics on the periphery, OBGYNs, who couldn't otherwise do IUIs, they are being enabled to do IUIs. And that changes significantly the process and standardizes it, similar to a very experienced lab, because all we need is a chip and a small $100, $200 incubator. So that kind of has a huge impact. We commercialized this technology first in Turkey, and that's where that paper came from. And okay, no problem. So that kind of has a huge impact, because in really small villages in the east of the country where there are OBGYNs, now a lot of these poor farmers don't have to sell their land, sell their tractor, or sell whatever they had to kind of come to the big city and pay the big bucks to do the ICSI, and we're seeing a lot of good success. And to highlight the importance of this work, and I mentioned that anything that's up to the blue line is things that you could see under a microscope. But bringing this tool, we could then pick sperm for low ROS, low DNA fragmentation, and with Palermo's paper, now we're able to pick for lower euploidy rates that's going to impact the embryo. And that's, I think, really exciting to see what's going to come out of it, because this is only clinical in the past 5 years now, which is wiping out the centrifuge as a tool in the fertility clinics. And I saw this, coming commonplace. I saw it in a fashion magazine, which I wasn't expecting, just randomly. And one of the clinicians in LA area, which I've never met, was saying that this was a huge game changer for them, and they only charged almost $400, thinking that this process is about $30,000 to $50,000. The cost is minimal, but the impact is significant. I would like to acknowledge some of my collaborators on the clinical side, starting with Brigham and Women's Hospital, George Mutter, and it was all their effort and interactions that made this tool to go and appear on Scientific American as among the standard lab tools. It wasn't an article on the Zymo tool or the tool I described, but they just considered it to be a common tool that everybody uses in a fertility lab, like a plastic dish, which is music to my ears. So I want to go back to talking about the understanding of the sperm cell almost as a micromotor led to us really change a field, and it's still continuing. And I want to show you how we approach this ability to bring fluidics and control over fluidics into changing and impacting other fields. And this is as early as 2007, and I see Tom Boland in the crowd, who was the first publication on printing cells using inkjet. And over the years, that field has grown, And we have developed a series of different acoustic and robotic-based controlling methodologies to really assemble things, assemble cells, build constructs. And this was in 2007. I had a postdoc, Dr. Sang-Jun Moon, and who's now a very successful professor in Digest in Korea. He came and drew this, and he said he wanted to build this, and we were discussing what would be the best printer to build. And out of this drawing, we built this in 2007 and started printing cells by an ejector. That's where the ejection setup is marked by generating signal processors. And it's pretty much a simple idea of droplets that are encapsulating cells in hydrogels, ejecting droplets, and we are able to control when the ejection is going to happen. That leads to, and by using UV or heat, you can cross-link them and build multi-layer, multi-cellular, or multi-material constructs. And that idea was great only by controlling the droplet and cross-linking. I started collaborating with Metin Sitti, who's now a director at the Max Planck in Stuttgart. He was at CMU at the time. And we started building these hydrogels, which is actually these microhydrogels are pioneered by Ali's work in the field around that time. So we made these different hydrogels and showed that by bringing an 8-axis robot, we could actually play Tetris with it or assemble it in any way and shape and format we could do it, one gel at a time. And we were interested in studying different cell-cell interactions at the time, especially between cancer cells and fibroblasts, working with Tayyip Hassan. And you could kind of make these different designs. The problem there was we were just changing one thing at a time, and we wanted to assemble things really rapidly, because if you look at cardiac tissue, you have millions of cells in a very small packed area. How can we bring cells and even eliminate the gel so that it can be a scaffold-free design? And that's where we started thinking about acoustics. And Faraday's cage is a known concept where you kind of get no electromagnetic signals into a conducting cage. But Faraday worked on all sorts of ways, and these waves on the surface of your cup are known as, on the surface between the air and the fluid is known and defined as Faraday waves, and they have all sorts of beautiful patterns and resonances if you can control them. And we kind of use this concept to create a tool. It's just a simple channel where we could put micro objects, design previously how fast we were going to shake them, and based on that shaking, you could create all sorts of patterns, and this video is in real time, and you could redesign and reschedule and build all sorts of different shapes and forms that are within the resonance of the Faraday wave for that geometrical structure. And this led to work with Dr. Puchan. Puchan was a postdoc in my lab, and he kind of engineered this work, and you'll see at the time, if the second video plays, you kind of see very clearly the resonances on the surface that assembles and reassembles. And once you assemble them, you can apply, change heat or UV or use a fibrogen to make the surrounding gel, so the cells are packed together and they can communicate sooner. And a group in Switzerland, in Davos, is trying to commercialize this, and I'm trying to help them to do it, to see if they could make a good printer out of it. Our interest has moved on. Since we can make these really thick cellular matrices, we built cardiomyocytes that are assembled, and these are iPS cells that we obtained from Dr. Joe Wu's lab at Stanford. And then we built them in a way that they could, let's see if the videos are going to play. Yep, that's great. So we built them in a way that we could put the ring or the toroid shape in a plastic that we could control the shaking with electrical signal, and that would almost be a micro swimmer by just engineered by the movement of the cardiac cells on this toroid or the cardiac belt that we built by this system. And it's very powerful because everything that in that toroid is just consisting of cells. Our interest in these micro-robotics, and I'll tell you at the end where we are trying to go with all this control and micro-robotics and how it gets very interesting at the GI tract and cancer. And I'm going to start moving a little faster to allow that time. We started taking cells and building small cellular robots. We wanted to control where they go and how they move. Kind of see that when you have this, again, controlled magnetic field, you can control where these things are actually precisely going. We used live objects such as live bacteria, and in this case, this is a really interesting, Volvox is a very interesting light photosynthesis making fungus, where we can use it both to create mixing, we can tie it with certain nanoparticles for imaging, and we can use these nanoparticles as a heating mechanism. And we've been trying to see if we could inject Volvox into different locations and tumors in mice so that we can use all these four effects at the same time and achieve tumor shrinkage. And this was, I think, published in ACS Nano the previous year. And do we imagine Volvox being entered into the human solid tumors at any point? Maybe not right away, but there can be other types of bacteria that we already have in our body that we can imagine that we would use. So this all robotic world ties to this one interest that we have now in the lab, which is the GI tract. And when you look at Google Maps, you can actually really resolve where a single spot is. And we can do the same if you look at the evolution of geography from Ptolemy all the way to Google Maps. You see the advance that has happened. And here you're looking at Da Vinci's sketch in 1500s, and going all the way to today, we have imaging tools. We can image where things are, but we don't know where things are being excreted from. If you look at the GI tract of 30 feet long organ with different pHs, with different chemicals being excreted, we still don't have a map of different diseases taking place at a very simple location. I'm not only talking about cancer, but other diseases that are important that lead. And this was a very nice review by Bob Langer's lab that actually studied and described different locations in the GI tract and how it could be important. But the key aspect of this is that we are not really fully informed by just knowing where there is a marker, but we want to know where it is coming from, because that's where we can understand where the disease is coming from. And our specific interest is trying to go build a system that could go and to different locations and sniff out different chemicals, the microenvironment, and do it in a way that we can integrate it with some of the existing technologies. And that brings us to integrate it with imaging, but we want to do it in a way that it could be just something that you can ingest and walk around during the day, and it can be just collecting samples, and analysis maybe is done outside, maybe analysis done on, depending on the functionality. And our work has led to a lot of interest in the extracellular vesicle space. And we are really interested in EVs with this collection system because EVs carry RNAs. If you think about the GI tract, you can get access to microbiota, you can get access to different chemicals when you are maybe interested in stool samples, but that doesn't give you the localized information. And RNA degrades really fast. You can get access to DNA, but RNA is really hard to get a hold of, whereas vesicles are able to actually preserve the RNA so that we can get functional information, functional biochemical information. To this end, I'll skip these slides talking about extracellular vesicles because I think this crowd knows what we are talking about. I'll just suffice to say that they carry everything a cell carries, from proteins to lipids to DNA to RNA, and cancer cells produce more of these. In different cases, of course, it might change from cancer to cancer. We still don't have really a good tool, a standardized tool, except maybe ultracentrifuge. Again, going back to the centrifuge story, to isolate these vesicles. But if we could have a functional tool that can control what size ranges we are extracting, what specific markers we are extracting, and doing it in a very high yield way, we would be in a very different plane to study this. So we invented around 2017 a device that we call exotic exosome total isolation chip, compared it especially with low volume plasma samples. Every time we go to our cancer researchers, we are told that they have precious samples, but they can only give us 100 microliters of it. And there is really no way of getting enough RNA or proteomics out of that small samples to enable enough downstream analyses on the omics side to really do discovery and understanding. So we specifically designed this device for high yield, and It's being translated and commercialized now by Mercury Biosciences, which is a GI company. And hopefully maybe in a few years you might change your mind on ultracentrifuge and use this device. But the idea is we can compartmentalize the system in a way that we can get different exosome populations in one shot. And the reason we want to do that is exosomes are being created, are originating biologically differently from the cell. Either they're being shed out or formed inside the cell, and their cargo changes. And it's a function of what they encapsulate. We have quite a few publications, which I won't get into a lot of them, on different applications of exosomes from the cardiac space to tissue engineering and regenerative medicine to cancer. But I just want to highlight one thing that's relevant to exosomes before I kind of go to the last stage of my talk. By looking at the exosomes, we can have information about their origin, meaning which cell they might be coming from. And this was a paper that was investigating that in differentiating iPSC cardiomyocytes with Joe Wu. And when you look at exosomes, they have an interesting aspect that they can actually go through the blood-brain barrier. And I chose two cancers that are actually being clinically studied now in my lab, specifically brain-related diseases. The reason for that is, imagine how much you can learn from the brain tissue and what it's been going over the blood-brain barrier into circulation using exosomes. If you could really go to a location and control where this device is sampling and sniffing from, right at the location that is the proximity of the tumor or proximity of the disease, how much more specific information that you would be able to get. So that's kind of the logic and thinking here. What I'm showing here, and this is not a glioblastoma talk, so I won't dig deep, but in glioblastoma, we would like to know if we have a disease that is aggressive or not, and the only way to do that today is by actually drilling a hole into the head and getting a biopsy. Could we do it without, could we do the diagnosis without actually having to go through surgery? And some of the initial work that we are doing with glioblastoma patient plasma samples, and I work with Dr. Solarolo and Dr. Piteri at Stanford, and Dr. Solarolo is at the Koç University, who's a neurosurgeon working on these questions and samples. We can get these extracellular vesicles, study them, confirm that they are vesicles, and then, going back, wrong slide. And going back, if you look at the two cases of meningioma, which you don't necessarily perform surgery and just keep watching it, versus glioblastoma, which is an aggressive cancer, if you could identify it by looking at omic markers, then you wouldn't really necessarily have to do either for diagnosis a biopsy, or you would be able to monitor the disease non-invasively. Another area that we work with Dr. Michael Zinnia, again from Stanford Radiology and RSL, can we look at the brain tissue of Alzheimer patients and rank them based on their iron content, because iron correlates with Alzheimer's, and Michael has been studying this for many years. Can we pull vesicles out of these samples and look at their characteristics? So can we stage Alzheimer's by looking at initially the tissue, and then can we carry it forward? So those are the questions that we have been looking into and identifying differences between healthy brain samples versus low and high Alzheimer levels. But these kinds of changes and looking at specific biomarkers, that's our mission pretty much, to change how the diagnosis is done and reduce non-invasive procedures. Now, taking couple steps to tie to the concept, if we can learn so much about brain through plasma, through tissue, could we create a system where we could have it travel, sniff out and collect effectively from every location that we want, and collect samples? And for the sake of time, some of the slides I'll start going faster. Some of this is published, so I will not spend too much time just to show you guys what we have. Our initial thought was, again, a microsystem that could open up selectively based on pH out of a pill and then sniff these vesicles out. As I showed you, we have the capability to collect enough RNA and proteomic information from just 100 microliters of sample, and we built pill-sized robotic systems that we can control that will allow us to go different locations in this system. And I mark them as 1, 2, 3 because the pH in the stomach drops significantly as you kind of get out of it to the small intestine, and where the duodenum is where pancreatic juices go in. And I talked about pancreatic cancer earlier, so one can connect. So can we go through the same system and assemble things? And can we do it with the hand of hydrogels to really absorb samples and retain them? And that's some of the biomaterials work that we have been doing for these systems and trying to optimize what we collect. We are not as much interested in collecting the bacteria because there's all sorts of bacteria in a microenvironment, and what really matters are which ones are active. And there's a lot of bacterial exosomal communication that we would like to actually capture here. We are mostly interested in those type of biomarkers because that will tell you more, give you a better picture of what the source of the disease is. And you can also capture bacteria with these tools, as others actually have reported some of the work. And we have been optimizing the magnetic control of these systems, as you see in these videos. Let's see if they're going to play. We are trying to find surfaces that are rough, and we are going to try to maybe collect and control multiple of these pills at the same time, and so that we can actually, somebody's really excited about my talk, giving alarms to the whole state, and then tracing it, because GI tract is not just a stable organ. There is a lot of fluid movement, and that's kind of what we are trying to try with different flow rates to kind of keep and retain a robot at a certain location. And these optimizations led to papers, and we are now in the phases of figuring out building something that we could try in large animal studies. But the idea overall, the mission is through these different tracks and diseases, can we create an atlas? And when I say an atlas, not something that you can see with imaging, but a biochemical and a secretome atlas of the track. And it's a very hard problem to address. And as we are doing it, how would this translate all the point from creating this atlas to it becoming a biomarker system and a point of care diagnostic medicine system? And I agree with the alarm that this could actually change the environment, the global environment. Think about global health. I worked on HIV for many years, and today the biggest problem, the biggest killer, as we know, is HIV, but cervical cancer is huge. If you could trace some of these cancers by a pill that people could use and send out to an outside location, you could change how we are doing healthcare in a preventative manner. And our lab, I credit a lot of this work to Fernando Soto, who actually has done his PhD in UCSD with Albert Wang, and Albert is here. And Fernando has been doing a lot of this work, and he's a very well-trained PhD student, so thank you, Albert, on behalf of myself and Fernando. And we've been trying to kind of see how we can push these things to the clinic. And one thing I want to really underline as I'm finishing my talk is these tools and instruments, they give you a chance to look at differently to disease. As we are doing that, we are very interested in disseminating the work we do, and also mentoring and sharing the information with people who are coming from very diverse backgrounds. And that's a global statement. And I would like to thank all my lab members and funding sources and Stanford who have made this work over the years. And I would like to thank you for the opportunity to be here today, and happy to answer any questions.
Thank you very much for your talk. These extracellular vesicles, they are really small. So besides sorting them by size, do you envision any other type of technology that is required to analyze them in a high throughput manner?
So I guess our goal is to take these samples, selectively pull them out, and then where the real information comes, once you have enough material, enough lipids, enough proteins, enough RNA. What really comes in is the next phase of omics. We do mass spec. You can do other more directed Luminix type approaches if you know what you're looking for. If you're looking for, let's say, an immune panel, that will give you more targeted information. Or RNA sequencing actually is really useful for the kind of broad things that we are looking for. And once we identify certain genes and targets, we go with PCR to kind of confirm that they are there. And for proteins, we do the Western blotting. So there's many sets of rules after the isolation where actually the biological and bioinformatic information starts. And that's what we are trying to get to with the right sample collection with the instrument, and then with the right and repeatable, reproducible sample isolation, and comes the downstream. So we're trying to kind of bring everything all the way to downstream in a standardized and successful manner where all the investigation starts. Thank you. I know that when I threw too many things to the crowd, I get silence, so sorry about that. Thank you very much.